Hey everybody, welcome back to another exciting episode of Bio6 Garage Lab. Today in the garage, we're going to be doing a Bradford assay for protein. This is a standard assay which is used in research labs all around the world. So this isn't something that we've modified for teaching. This is done in research labs. And what it involves is the use of a reagent called Bradford reagent. We're also going to use a diluent, which is going to be 0.15 molar sodium chloride. We're going to be working with a 0.4% bovine serum albumin, BSA protein standard. We're going to use that to create a serial dilution and make a standard curve. And then we've got ourselves 3 unknown solutions of protein. In addition to this, we're going to be using some 2 ml microfuge tubes to make our dilutions. Don't worry, the colors don't mean anything, they're just pretty. And then we are going to measure the protein assay using a spectrophotometer. So we've got ourselves our spec cuvettes. The first thing we're going to do is we are going to add 1.5 mils of our diluent, our sodium chloride, to each one of those tubes. We're going to use a P1000 pipette, and we're going to dial that pipette to 750 microliters, and we're simply going to pipette it twice into each tube. So I'll show you how to do the first couple ones. Now, because we are basically pipetting the exact same thing into each one of these tubes, we don't need to worry about changing our tips. So I'll do a couple of these, and then I'll finish this up, and we will come back and we will do our dilution of our BSA standard in order to create a standard curve. Okay guys, the next thing we're going to do is we're going to create a series of serial dilutions of this BSA standard, so that we can measure it in a spectrophotometer and create a standard curve. So to do this, we are going to add 500 microliters to our 1.5 mils of of sodium chloride. So we're going to add the BSA here to our very first one. We're going to shut this tube, and we're going to basically invert it up and down. Give it a good mix. Get yourself a new tip. Now, you're going to take 500 microliters of that, and you're going to pipette it into the next tube. Close it up, give it a mix, get a new tip for your pipette, and keep going down the line. Now, what we're doing is we are making 2 mils total, of which 500 microliters is the BSA, and that's 2,000 divided by 500, and I think that's a 4-fold dilution, isn't it? So I'll complete the rest of the dilution, and then we'll move on from there. Okay, we've finished our serial dilution. We have our tubes. We've got basically the first 7 of them have 1.5 mils in it, and the last one has 2 mils. So all I'll do to even things up is I will take out 500 microliters from this tube and dispose of it. And we now have every one of our tubes with 1.5 mils of diluted BSA in it. I'm going to add one more tube to this. It's going to be the blank that we're going to use, and it is going to be simply 1.5 mils of our sodium chloride. Okay guys, here comes the fun part, where we get to add the reagent and we get to watch some pretty color changes. For this, we are going to add 500 microliters of Bradford reagent to each of these tubes. The first one I'll do is going to be our blank. And our blank is just 1.5 mils of water and Bradford reagent. It's kind of hard to tell actually in this colored tube, but it's sort of a lightish blue color. But watch what happens now when I add Bradford reagent to our first solution, which has the most amount of BSA. Give it a little inversion, and now you can see it's got a nice blue color to it. So I'm going to keep doing that. I'm going to keep changing my tips, using a new tip, and adding 0.5 mils or 500 microliters to each of these tubes. I'm going to close them all up, shake them all up, and then I'm going to add them to our spec cuvettes, put them into our spectrophotometer, and measure up the absorbance, or basically measure the concentration of that blue color. From that standard curve, we'll be able to plot out that standard curve, and then we'll be able to measure unknown protein concentrations and determine their actual BSA concentration. So there we go. We're going to add the last one up, and then I'm going to close each of these tubes. I'm going to shake them up and then transfer them into the cuvettes. So the total we have in each one of these microfuge tubes is 2 mils. So if I set my P1000 to 1000 microliters and just simply pipette twice, I've got my 2 mils. First thing I'm going to do is I'm going to do my water. This is my blank. I'm going to pipette it over into this first cuvette that's kind of off to the side so I won't get mixed up. You don't want to actually write on your spec cuvettes. Yeah, and I could eject the tip and get a new one, but you know, it's water. But it's always good practice to eject your tip if you're not sure whether you've contaminated it. Now I'm going to show you a trick, guys. I'm going to start with the lowest concentration, and I'm going to pipette that into my cuvette. And because I am going up in concentration, I can use the same tip. I'm going to pipette that in there. Transfer this one. And I'm going to keep going all the way until I get my most concentrated solution. And that way I can keep using the same tip. If I started off with the most concentrated solution and worked my way down, I'd actually have to change tips. I'm also making sure that there really isn't a lot of liquid in that tip when I'm done. And if there is, I can use the hard stop of a pipette in order to get rid of that excess liquid. So I can basically use a hard stop, and you'll see I have no liquid in the tip. That's another way to prevent contamination. So we're almost done here. And you can see actually the colors are getting a little stronger, aren't they, as we move along. And here's our last one, which is going to be the most concentrated of our BSA. So there we go. We've got everything done. Let me show you basically what these colors look like, because it's kind of hard to see from an angle. There is our serial dilution. You can see at the very first dilution, we're nice dark blue, and our blue gets a little less concentrated as we move over to our most dilute solution. You'll notice I've got 3 empty cuvettes here. These are going to be for the setting up of our unknowns. So we're going to set those up now. So now we're going to set up our unknowns. We've got 3 unknowns, and we're going to set it up with the same recipe that we had for our known BSA. We are going to take 1.5 mils of our diluent, our sodium chloride. We're going to add it to each one of these microfuge tubes. We're going to add 500 microliters of each one of these unknown protein to that. Okay. And then I'm going to change my tips in between each one. So here's protein solution A. Change your tip. Here's protein solution B, because I don't want to contaminate. And then we'll do protein solution C. Now, there's not enough room in this tube to add our Bradford agent, because remember, in our serial dilutions, we had 1.5 mils of sodium chloride protein mixture. And then we added 500 microliters of Bradford. So how are we going to deal with that? Well, it's simple. We're just going to remove 500 microliters of this BSA sodium chloride mixture. So I'm just going to take out 500 microliters from this one, 500 microliters from protein B, unknown protein B, and 500 microliters from unknown protein C. And now I basically have the same thing, don't I? Now I can add my Bradford reagent. And I'm going to add it such that I don't touch the tip to the solution, so I don't have to change my pipette tips. If I were to suddenly do this and contaminate the tip, I would simply change out my tip. Tips are only worth pennies, and it's not worth contaminating your experiment. So when in doubt, throw the tip out. And we now have our full set. We've got our blank, we've got our serial dilutions, and we've got our 3 unknowns in gorgeous colors of blue. We're now going to measure the absorbance of these materials using our spectrophotometer. All right, here's our Ward's visible spectrophotometer. Again, we've used this before in our diffusion lab, but let's remind each other what's involved. We've got ourselves the sample holder. The sample holder will allow us to measure 5 samples at a time. The top sample is going to be where we're going to put our water blank, and the remaining samples are going to be for our experimental samples. And again, we just simply push this back and forth in order to move the cuvette into line with the visible light, which is going to come out right where my finger is there. So that's our sample holder. You notice I even drew on it to remind ourselves what's what. Here is our wavelength indicator. We are going to be going for 595 on this. So there is about 590, there's about 595 right there. Again, we've got ourselves the buttons, the mode, we'll make sure we're on absorbance. This is our blank button, and the other 2 buttons we're just going to ignore. So the next thing we're going to do now is we're going to load our blank and the first 3 samples of our serial dilution in there. So I've put the blank in here. We're going to take our most concentrated, we're going to put it in, and again, there is an orientation to this. You can, I don't know if you can see it, but there's a little arrow there. But you basically put the cuvettes in so that the arrow faces the direction of the light bulb. So we put them all in the same way. These are our top 3. Right there. We're going to shut the lid. Make sure we're all the way out. We've pulled it all the way out. We're now going to shut the lid, and we are now going to blank the machine. So we're going to make sure we're on absorbance. We're now going to go to the 0, 100% T, press that, and it's going to blank. If it doesn't blink, press the button again. There you go. It's not blanking unless it's blinking. All right, next thing we do is we're simply going to push our lever in 1 slot at a time. So here comes our number 1 sample, 0.570. Our next sample beyond that should be lower, 0.508, and the next dilution sample should be lower than that, 0.395. All we have to do now is simply remove the bottom 3. So we're going to pull this lever back out. We're going to keep the blank where it is, and we are going to remove the bottom 3 samples in slots 1, 2, and 3, and we are simply going to load the other ones in. Keep the blank there and load in your other samples. So there we go. And again, make sure they are all in the same way. There they all are. We shut our lid. We push again. We don't have to push anything yet. Notice we have our blank. Now the next thing we do is we're going to put 1 in, and we're going to read that. That's going to be 0.225. Go in again. 0.084. Go in again. 0.028. And we're going to keep doing that until we finish up our standard, until we finish up our serial dilutions. The serial dilution data is going to be online on your Canvas page. The last part of this experiment is to do the unknowns. So we've got our blank, we've got our 3 unknowns in there, and all we need to do is read. So again, we just push the lever, there's an unknown, and then we simply do that for the remaining 2 unknowns. And there's the last unknown. So having done that, you've completed the experiment. Now all you have to do is take the data, which is loaded up onto your Canvas page, plot your serial dilutions, determine the molar absorptivity, which is going to be the slope of your graph, and then come back and apply that equation to the Beer's law equation for your unknowns. And you'll be able to determine the concentration of your unknowns. So that is yet another episode of Bio 6 Garage Lab.